let's start with Kanzu as a second one, and then I'll come back to somebody who's present in the room and then go back to stuff online. And then just go back and forth to just make sure everybody who's present. So just get ready to prepare for lots of holes to be put in your FRDPARRCs. So now this is really rubber meets the road. So we're going to be thinking a lot in terms of what is a practical, very pragmatic approach, but still that fits the constraints. And where is the heart of the problem? I think that's really what the discussions is. At this moment, you need to understand where is the heart of the problem. Because overall, once you've cracked the hardest puzzle or where you felt was a key intellectual bottleneck, we can then go back and build a system around it. But what is it that would stand out in terms of thinking about a solution? Why is it that we are pursuing it? What is the composition in the team, and what skills you guys already have or might acquire quickly to be able to crack? So I think as you've been building a FRDPARRC, the idea is to have it to be very comprehensive, and you might almost first be tackling one single row, for example, in the FRDPARRC. That's going to be one of the goals. So we're going to turn to the OSMI bags. You guys can either draw on board, or if you have, just join on Zoom and then you can just share screen. Which way do you prefer? Zoom or board? This is like Chalk Talk style. So yeah, why don't you join Zoom and then share your FRDPARRC and walk through it. And then just so everybody is aware, we discussed this problem Thursday, I think. Here is one of those bags. These are the OSMI bags. And one of the key discussions we really had was around the material science for these bags to really make sure, why do they leak? How do you deal with this from a manufacturing point of view, and thinking a lot more about flat manufacturing for something like this, and try to find a pinch point around if there are some key failure points in this design, where would you want to approach this from? So that's kind of where we were on the discussion. And maybe one thing we'll try to do is discuss a little bit about what the next steps might be for the team. And then the expectation is that everybody is participating in the discussions for every team. So although in your normal time for the class, you're just thinking about your project, but in these brainstorming discussions, everybody should focus on thinking about the team that's presenting. This is your way of providing ideas and feedback to other teams. Okay, I see Ivana is already online, so then you can just share screen. Okay, take it away. And then it's not about very comprehensively presenting your FRDPARRC because people can read it. It's about, oh, this is a thing that I really want to spend time discussing because we're leaning towards it. And it might imply that that's where our first sets of efforts might go. So we just started we're just going to be running this, everybody presenting FRDPARRCs much more in the context of the analysis layer that's going to come. And this is run much more as a brainstorming. So anybody can jump in anytime, clarifications, questions.
Can people hear us from
online? Can somebody give a thumbs up if you can hear Ivana? Not me.
Okay. Yes, thank you. Yeah, so we discussed a little bit about this, and we started like mapping out a little bit what are the problems within ostomy bags that we think we could tackle, or that are, well, the main ones, and then we can zoom in. So the first one really, it's like the cost, what we explained the other day, no, that there are a lot of people having access to surgeries, and it's increasing every year. And less people having access to ostomy bags. But from there's even with the current solutions, there are problems as well.
So just a cost, which country have you guys chosen as your key analysis country? And then what's the primary cost currently on the availability?
So, we have been exploring India, since one of the team members is actually in India, and he's been
And
having a look at the
Is that
cost.
Webhub?
Yeah.
Okay, yeah. Webhub, are you online by any chance? No. Okay, yeah.
Yeah. So he had a look a little bit and he told us he managed to speak to a manufacturer actually. And he said that the cost of actually building these bags is super low. It's like in China and India, you can build it for $50. But that then the supply chain, it's like very complex and then there are a lot of margins also from the company. So basically that's why it's so expensive.
How much do they sell it at currently in India?
Mm,
Like just orders of magnitude. Should I think of it as $1 per bag, $10 per
I
bag?
think he said like $40 per month.
Month. So that would be
Yeah, it's $1.
$1.50.
Yeah.
Okay, so 50%.
Professor, could I pitch in?
Yeah, go ahead.
I'm not in the group, so there are two people named Weber in the class.
I
and
see.
I'm the other one. But the cost in India for one bag is around $2.
$2. Okay, yeah.
That's
Yeah.
the cheapest one.
Yeah, perfect.
Yeah, so there are like really big margins. But one thing we were worried about is to discover that it's actually quite cheap. I mean, not worried, but it's a good thing,
Yeah,
but
yeah.
there's so big margins,
Yeah.
so I don't know.
Yeah, I think the key thing is that, that $2 number sounds low, but you are paying that for life,
Yes.
right? And I think then if you look at, so the way you should do that calculation is much more driven by percentage of a household income. And then on an yearly income, what percentage goes in? And then you would start seeing that, oh, that's equal to how much money people might be spending, say, on food or something else. And then it starts to, and especially then you give this criteria to say, oh, our target market and our target demographics might be in these, many rural areas, the surgery is not available, so the numbers are low. Many urban areas, the per capita income might be a little bit higher. But there's this intermediate small towns where the surgeries are available, but that price doesn't match. And so then you can even narrow further, not India, but just you've chosen a sub-town and you could say, oh, here are the number of surgeries happening. And this is really where, and because even those, like, as you narrow that down further, you might realize that even then you're talking about millions of people.
Yes.
May I ask a question?
Absolutely. Jump in anytime. Go ahead.
I was wondering how often does one need to change these bag?
So normally it's once a day. Some people change it every 2 days. And there are two types of ostomy bags also, the ones that are like one piece, which means that it's all in one. And the ones that are two pieces. Two pieces means that this part is separated from the pouch. So when it's two pieces, sometimes the piece that is in contact with the skin. Well, if it's two pieces,
The exchange, you have to do it at the hospital, or that's something that the patient does it themselves?
They fix it at home. So basically the way it works is they have this system that they cut manually every day and trying to mimic the size and shape of their stomach, like cutting like an adhesive. They put it on the stomach here, and then there's a way of emptying that a lot of times per day they're gonna go to the bathroom and empty this, which is gonna be full. And then they close it like this, and once a day they would like throw it away and put a new one. But they, or anywhere they can.
Yeah,
Okay.
yep.
Oh, go ahead, Yuki.
I was just thinking about in terms of with like this seems to be prone infection. So maybe thinking about antiseptic sort of like in that area would be some nice feature to build in.
So when you said I guess, I mean, amongst all of the thread, this is on the summary side, is there something that's rising as a price is one, that makes very clear. Is something rising that is a critical problem that if you crack, it will have a tremendous impact on just the user experience. Like amongst many things, if you were to put, and this can be a little bit on a gut feeling that amongst all the people that you've spoken to, like if we could solve one amongst all the problems, which one would that be? Cost, of course, is one criteria that anything that we add as features, anything as we think about, this object is a history of innovation by many, many people to get to this point, but it's still not working. And so if you were to put your finger on one, which one would that be? Like in terms of the discussions that you guys have had, in terms of feeling like, oh, this is really a huge space for us to rethink.
One thing that is not working is leakage.
Yeah. Yeah.
Like it's leaking. 50% of people, it leaks every day or every week.
Yeah.
And it's burning the skin, and it's with these bags.
Yeah. So I think then the plan is in the FRDPARRC column, let's really focus on that functional requirement. Leakage, like amongst all the other things, and then making sure that that is compatible with the rest of the manufacturing pipeline itself. And then we can expand it both in terms of, is that leakage is really what we truly need, maybe is a completely new adhesive. That's one subsection. Another one we talked about is the conformality of the body. It doesn't make any sense to have a flat object be applied to something that's very conformal and folds in very strange ways. And that folding is a really beautiful problem to begin with. So now the plan is in that leakage row, let's write down on the board as many ideas. And again, this is now, because we can discuss the basics and then we'll just keep extracting more information from all of you as you've been thinking. But the idea is to just, if we just focus on leakage on the whiteboard, let's throw as many ideas as possible on how do you seal a conformal. So when I say a conformal, it's just, you can touch your own stomach. It folds in all kinds of funny ways. It's not flat. And you have this flat disc, which is also compliant. It's soft. And then you have there's the chemistry side of the story. There's the geometry side of the story. There's the amount of pressure. There is a notion in terms of thinking about what happens to adhesives as a function of time, right? I mean, as you all know, skin cells die when they can't breathe. Why is it that adhesives over time fall off? Like, what is it about adhesives? How do they change chemically? So let's just make a list there and just throw tons of, I don't know if there is a marker. So I don't know which one of you want to just jump on the board and let's just write down as many of these ideas. Yeah, I think the idea what is important is to just document as kind of quick things. And then everybody should just do rapid fire of ideas. And we can put them in different categories. But what we're trying to crack and solve is the leak. And then anybody online itself too. So I'm going to unshare this for a second, just so we can see. Oh, I can't do that. You have to do that. Yeah, so you can just write the ones that we just talked about. So there is the conformal geometry. So you could just write like geometry or something, or geometry of the stomach.
There's also that geometry of the actual.
Of the device, yeah. So then write geometry, and then you could do stomach meets sticky pad, right? What do you, is there a term for it? So we're using a technical term.
The stomach.
No, the disc that attaches, is there a name for it? Stroma. Okay.
So it's like this, the part that is like this, and then there's the pouch.
No, the ring-like structure that's soft and has the adhesive on, it's called the wafer. Okay, let's just call the wafer. Stomach wafer interface is what we're interested in.
Is it possible? I think
everybody will just be talking, so it's gonna be super hard.
yeah, I'll just put this closer to you.
Okay, Henry, go.
I just had a question. In typical, I guess, ostomy bag installation, is there something that stays in the stomach that interacts with the bag that you place, or Like, is it always just?
It's so you
That.
just have like a bit, I actually brought, you just have like your intestine that is like out of like, I have like this fake one, but it's like, this is like intestine and it's literally here, but it's from you, it's like your intestine. And then here it's where they cut this, but most don't look round. They are actually very complex in shape. And
So actually, this raises a really important question, that theoretically, there will always be a portion of skin that will remain exposed to many
Yes.
of the acids. Because no matter how precisely you cut it, there will be a margin. And even when you're cutting it manually, there will be all kinds of variable margin. So it's kind of an interesting thread that if the goal was though that there is never an interface between the skin and the acids that come out, that theoretically is very hard. We can, unless we come up with something totally different that mechanically interfaces with the soft tissue, which I could assume was a big no-no. Because the tissue properties of the intestine is so different, like skin is designed to take all kinds of other things, and putting an adhesive on that is okay. And then the other thread is, oh, what about the space of gels, like soft materials that remain fluid, like Vaseline, or again, that's a pretty tricky space because then suddenly we're entering space where anything could get inside. So I think, yeah, I don't know, how would you capture it? Just capture this as the fact that the wafer hole and the intestine by itself, they're two different geometries also. Like,
Yes.
it's just one on top of the other, there will be a ring of skin. Yeah, sorry, go ahead.
Well, it's okay. I think, I was wondering if you were to use something that's a little bit elastic and like have it just slightly smaller and then to put it on, is it bad for it to kind of add just a little bit of compression to the?
no, this is okay as long as it's very elastic, because it stays the same after 1 week of surgery. It stays like this forever. But when it removes output, it has a bit of movement. It moves a little bit. So anything
Yeah, I
that
mean,
moves
it's
in there-
peristalsis.
Yes.
Basically, that's what drives
Exactly.
most of it.
Yes.
But
So
then
it needs
peristalsis actually also implies that it opens and closes too.
Yes, a little bit.
So that by
It's
itself
like a
is-
little.
yeah. Yeah.
And actually, one thing that happens sometimes is that because it's not retained, some people have their intestine go out of their body a lot, and then they need to run to the hospital so that they put it back. But if there was a
So what is the interface on the surgery side between the intestine and the punctured hole in your skin? Like, are there stitches?
Yes,
To make
stitches.
sure that some portion of the intestine is stitched and those stitches remain for years?
I think at the beginning there are a lot of stitches I've seen,
Yeah.
but then at some point they remove them and it stays like this. But
So
that's
somehow the interface also fuses to the skin,
yes,
right?
yes, exactly.
This might be a silly question, but is there some way to like add something, like when you're doing that connection, something that can act as an interaction between the bag and the
Like a third connector.
Right.
Some kind of,
Yeah.
yeah, so let's just write that as a connector. Let's also just write the word gels there, as a, so there's gels, there is connector, there was another thread that had just come about, on, I think, is leakage related to the fact that the object that you're interfacing by itself moves? So it's not a fixed geometry to begin with. So the fact that the intestine itself has some compliance or movement internally, not just your own movement. Any other things that people want to just toss out? And this is the moment you can be as wild as possible. Also, folks online, it's useful to just type in your messages and then we can see. I see Meghna and then after that Vaibhav. But there was a comment here first. Yeah.
I guess like I have a question. Okay, this is kind of going into like the design of like this bag itself, right? Like it kind of like I'm looking at picture, it kind of just like dangles, right? Like, is there any way for you to just like change that bag design in a way such that it like maybe like wraps around your back so that you no longer have that like gravitational pull? Like,
Yeah.
have like some elastic, just like a tube, like.
No, this is a good point because one thing people complain is that even the bag, it goes on the leg and sometimes it touches the leg while people walk, and there are people that even have a permanent scar of the bag touching.
Yeah, so one connection between the two is because in the leakage space, there's a whole nother space of the bag design too. But it's also possible that because if it's partially filled, that leads to a tug. And in fact, the design of the bag is impacting the interface and the contact. So maybe one thread to think a little bit would be is just gravity. This if you just make another thread around gravity. And whether as the bag fills more, it's more likely to leak.
Yes.
I think we didn't say anything about pH and chemistry, so there should be another umbrella around the chemistry / pH. Because effectively the adhesive is also interacting with a very high pH.
Yes, exactly.
Do we know the pH number?
We can check. The idea is
I'm
that
assuming it changes depending
Yes,
on
on the
where cut, but like order of magnitude, how much?
We
It's
can
like
check at the
range of like
restaurant.
4 all the way up to 6, depending on where you are.
Where you cut it, so 4 to 6. So that's an important thing to think about, that, oh, adhesives that we are looking at, we have a chance of testing them at some, because, that's nothing. Band-Aid industry wouldn't care about it. Like if it's, do we know the chemistry of the exact adhesive? Have you guys been able to dig in and say what exact adhesive is being used currently?
We have the name, but
Yeah.
we haven't really looked.
Can you just pull up, do you know the name somewhere? Like, can you
It's
just pull up-
written
I
somewhere.
just want to see, one person in the team should really explicitly dive in and see what is this chemistry, what type of bond is it actually making? And then while we are searching, let's, I know Meghna and then Vaibhav, you guys had your hands raised.
It's acrylic-based adhesives,
Because
hydrocolloid adhesives.
Okay. So colloid adhesives are different. But yeah, I think dig in a little bit because this will become a big portion, and especially the explosion in biologically compatible adhesives. I mean, just as we are learning from marine life, incredible adhesives and especially programmable adhesives are starting to come, where it's incredible adhesive and one specific thing changes it. Because again, it's a hard problem. There are lots of adhesives like acrylic glue that if you put in, it will just completely bond, and that's not so useful. You want the bag to be removable.
Yes.
So in some sense, and then the other approach in the idea space is, oh, let's really design a belt that works so that there is no adhesive. Like
Yes,
the
that's
best
what you were
adhesive is no
saying.
adhesive. And then I think the boundary between this adhesive space is something that is easily removable. I know how much it hurts when I pull Band-Aids for my kids. They always hate it. And so you can say there is a tension in this problem also.
Yes.
So on the adhesives, there is this notion of, programmable removability. And I think just knowing more of that chemistry, you can think about whether these things can be removed.
And then the water is a problem also sometimes,
Say
like
that
when,
again?
Water, like waterproof.
I see,
Like
yeah.
one of the main issues on why they change it every day, it's because it's when they shower or when
I see, yeah.
or
That's,
sweating.
yeah, so just water and sweat. It's also right, sweat is a little different. Go ahead, Tanvi.
Totally stupid, but would a magnet work at the attachment point, or is that like just harmful? I don't know anything about.
Yeah, no, I think at this stage, it's important to write something and then explicitly rule that out. Like, just as we said, belts, magnets. There are now compliant magnets that can actually be printed inside a rubber itself. But then the problem is that it requires something on the other side, and then you have to have a biocompatible. But I think at this point, it's fair game. Imagine a compliant rubber that gets mounted on the inside when the surgery is done, especially for someone that's permanent. And there's a lot of space in that. I mean, some of you have already used it. Fridge magnets are actually micromagnets that are embedded in these little compliant rubber sheets that you put on your fridge. Okay, I think lots of people online had their hand. Meghna, did you want to make a comment, and then Vaibhav?
Yeah, I just wanted to add one thing. Basically, you were talking about if there is a possibility of a skin barrier that can be used. Usually, zinc oxide paste are given so as to avoid the skin maceration caused by the contents of the stoma. It's a common, that's when they change the bag, they usually apply the paste and then place them, keep the bag on, back on again. So that's one thing. Yeah, that's what I wanted to say. Yeah.
Yeah, that's a beautiful suggestion, especially in some sense, you are trying to protect the skin. And then I can also see the challenge with it, that if you put a zinc oxide as a cream and then you change, then you could see the issue with the adhesive not working as well with or without the cream. Right? I mean, on one hand, whenever you get a wound, you are told keep the wound open and air it. And I could see that site, as it's starting to get irritated, you have a tendency of not wanting to put something, but that's not an option. You cannot not eat.
Exactly.
So no matter how bad the skin gets,
You need
actually,
some-
this is a bigger problem in some way. That maybe what we're missing here is we're missing repeated injury. It's much more of a repeated long-term aspect of this that's a problem. Because once you get a kind of an irritation, you don't have a choice. You still have to put the bag in the next number of hours. I mean, sometimes when you get a wound, you give it a break. But there's no break here, which makes this incredibly complex.
It's like a
So-
vicious circle.
Yeah, it's a vicious circle.
Hmm.
So yeah, I think in this, we should write something along the sides of long-term prolonged injury rather than a short-term. So, and I wonder if there is another space where this has been thought about is actually in prosthetics.
Yeah.
So in prosthetics, when people put something on a stump, you get into the same situation, and then it becomes really irritating, and then people just stop wearing a prosthetic for some time. So I think, I mean, the prosthetics is, let's look into what have people done in prosthetics, and it's also skin injuries.
Because we've seen that in a lot of prosthetics when they input when they amputate the leg, even in the surgery, they already place something so that the prosthetic is gonna clip
Yeah.
very well.
Yeah,
But it's
but the
both
skin
very
irritation is tough,
different.
and that's why people then when they walk, they remove the leg,
Yes.
and then they don't wear it, even if they would like to, for
Mhm.
some time for it to heal.
Yes.
But I think here that's not an option,
No.
which kind of creates this. Imagine two bags. One is the one that you want to regularly use, but another type of a bag that is really designed around when you have gotten an injury and you want to give a certain site
Mhm.
of protection. And it's a different type of a bag. It's not designed for you to be able to walk around, doesn't have the same form factor of being hidden under a skin, but it lets
It
the
lets it
injury
heal.
site heal.
Mhm.
And maybe that's the one in which there is a direct interface with the intestine,
Mhm.
and it's a connector rather than a bag, with the goal of eventually going back to the bag.
Mhm.
Okay, we'll take one or two last comments, Vaibhav and Avinash, and then I want to switch to another team. But I think this is kind of what I wanted to do. It's like in your teams, you need to sit and throw 100. We didn't reach 100. I think we're still like 25 or 30. You need to get to a point where you feel like, oh, have I saturated my thinking around this?
Mhm.
And we clearly haven't. And I mean, of course, you don't need the big group to do it. You need to do this internally. So then we're looking at these sheets. I feel like you've chosen a problem, you're really thinking hard about it, and there are lots of options to go. And then here is the list of, I can immediately see lots of things that you can do now to rule out and scratch to say, nah, magnets was a really fun idea, it's super nice, but a surgeon has to change something.
Mhm.
Too hard a road to climb. Let's, put it on a Okay, Vaibhav and then Avinash.
Yeah, Professor, I was thinking that for the leakage
Call
proof,
me Manu, Vaibhav. Everybody just calls me Manu. Yeah, go ahead.
yes,
Yeah.
yes, Manu.
Yes.
Ivana had mentioned last time that the stoma itself doesn't have nerve ending, so people don't feel pain because of it. So maybe to come up with something that is a one fit all kind, we can have something inspired by drawstrings. So we could tighten the surface around the stroma itself.
Mhm. Yeah, I think this is a little bit related to what in the connector space, that the connector has both compliance and different mechanisms. So either drawstrings. I think one thing to worry a little bit about, and again, just it's going back to writing the material properties of this. So you guys should know the skin compliance, and you should know the material properties of the wound site of an intestine. Because if it's very, very soft, tension cannot be used as a mechanism, because you could completely close it too, right? Because unlike other materials, it is, my intuition is that it's soft.
Yes, it's.
And although there is muscles, but let's put some numbers on that too, just so you feel. Let's go back just on that connector. It occurs to me, I know when people get, say, a certain cancerous lesion in the colon and other places, that they cut a chunk of the intestine and then they join it. So what do they use for that? I mean, sometimes you can just say, oh, it's long and I can still join. Has there been a material in surgery that's been invented to bridge a gap between intestine? So there might be a space where people have already thought about a material that interfaces with the open intestine, just as a connector in surgery, but that material itself could be viable. I mean, in arteries, for example, it's a little bit tricky. Stents act as a support, and that's also not a crazy idea. Again, but it really, and I think one thing that I'm seeing here is materials and chemistry, and just focusing on that port. Although there is a lot of other really fun, low-hanging fruit that we could do on the design, they're not hard. All of them are easy, and they will be incremental improvements. I think if you crack this in a fundamentally different way, it completely changes. So I think this is also another important thing to understand, is that you're not choosing the easy change, you're choosing the hard change, even knowing fully well that it might fail, or in a short time you can't deliver. I would much rather have us work on a hard problem and fail, rather than an easy problem that anybody else could have done.
Yes.
Then there's industries whose job it is to prove this, and maybe they're stuck somewhere. So I think this is another criteria we want to apply. So I think I'm gonna switch. Oh, Avinash, go ahead. One last comment, and then let's move to the.
Yeah. I was just thinking that how long one bag we will be using.
Just
Uh,
1 day or less.
yeah. So like how long will it be used? So maybe that is something which will, like,
Yeah.
the material we are choosing
The
will
leakage has like 12 to
depend on.
24 hours. Let's put a definition. And another way of solving this problem is, imagine making the bag so damn cheap that you
You can
find it okay to change every 6 hours or every 8 hours. And that actually reverses the problem in an inch. It solves the problem in a totally different way. It just says that, oh, I don't care about the long term. Yeah, that's a really good point, Avinash, because it changes the contact point, which has the possibility of leakages and interactions.
Yes.
And, I think it is interesting that reducing cost could change that paradigm in some way, and that maybe people are using it longer because of,
Yes.
insulin is a great example. When people ration insulin, it's bad. Like, the right answer for that is not figuring out some rationing protocol, it's to just make insulin cheaper. Okay, I want to switch. This was really fun, but I want to be fair to other teams. So Yuki, Dave, just take a picture. Take it away. You guys, I'll ask them to unshare, and you can share. And somebody in the team who wants to volunteer to be a scribbler for their team, because I'm assuming you don't have anybody in your team that's present in this room, right? Can you just
Nope.
say the team member names? Dave, Yuki, who else is?
Yeah. We also have Murnal, Meghna, and I'm sorry for pronunciation, and Divya is also part of the team, but isn't able to join.
Awesome. Okay. So who wants to be the scribbler? Can anybody volunteer? Yeah. Because I think, yeah, it's kind of fun to, did you take a picture, right?
Yes.
Okay. Otherwise, all of that is gone.
And for the ostomy team, there's some more comments the notes that were put in the comments here that might be helpful.
Copy the chat, because the moment we close the session, the chat disappears. So you guys should copy the chat.
Okay.
Okay, so if you unshare, the next team can share. How are we doing on time?
Oh, okay. Here,
Micro.
Okay.
okay, can everyone hear me okay?
Yes.
All right, so I'm just going to give about just a little three-side overview of Konzo disease, and I'll just point out by saying there's a lot of unknowns with
Yeah.
this, so
And
this
then
will be a
Dave,
real broad brainstorming session.
Since you've been to Congo a couple of times, just also do a one-line intro for yourself too.
Yes, for sure. Yeah, was previously at Stanford and just started a faculty role at the University of Nevada. I am very interested in infectious disease and the connections between immune responses and other diseases, which is how I got interested in this disease. A close collaborator that I was working on has been studying this disease for some time. So just as a bit of a background, and we did travel and interact with some of the folks affected by this. So Konzo disease is a sudden onset, symmetric, non-progressive, irreversible spastic paraparesis or quadriparesis, so like a paralysis that happens. It was first reported in what is currently Democratic Republic of Congo. It results in this stiff leg syndrome, difficulty of moving, and has different levels of severity. The word Konzo translates to tied legs. And interestingly, this occurs in an outbreak fashion. And here's a video that shows some of the different stages. So this is the first stage of disease. And again, it's non-progressive. So you end up with being stuck at one of these stages if you're affected. So that's an individual, the first one who could move unassisted. Second stage is being able to move, but still assisted. And the third stage is not being able to stand and basically being burdened, being restricted to being on the ground. So the burden of disease is especially high in Central Africa, and again, especially high in the current Democratic Republic of Congo. It appears to be associated with food insecurity, with lack of water, and famine and drought conditions. And without going into very much more detail, the pathogenesis of the disease is connected with consumption of cassava. So cassava is consumed by millions of people around the world, but it contains cyanogenic glycosides or compounds that can release cyanide. And if not processed properly, those concentrations can be high enough, it is thought, to cause disease in some cases, and this is one of the diseases that is associated with that. So there are many different aspects to this problem. There's a lot of unknowns about how it's about the factors that cause it to occur. There are inputs that we could, or there are ways that we could think of addressing this on the host side, like in the individual side, on the consumption of cassava side, on different ways to alter the processing of cassava, different ways to alter the growing of cassava, or different ways to approach education. So I'll leave it there to the rest of the team that can go through the FRDPARRC, if I'm pronouncing that properly.
Yeah. Dave, just before the transition, can you say a word about, is there an animal model system that anybody has shown that in mice or any other primate you could actually really
not very effectively. I'm unaware of primate studies. There is not a clear animal model for being able to recapitulate the neurotoxicity of this in terms of a similar kind of disability that you can induce in the animal models.
Okay. Yeah.
Yeah.
Okay. Let's jump to the FRDPARRC. And then I think the idea is the same. Although there is a broad FRDPARRC for the purpose of the course, and again, the team, you guys could, have a aha moment tomorrow and say, oh, in the class I said leakage was the biggest problem. Now I've realized it's not. That's totally fine. Then you jump in. But just it is about that really razor sharp, narrow focus on a sub-problem. So you guys can choose a sub-problem, but it doesn't have to be that today what you discuss as a sub-problem is the sub-problem, because that could change as you dig in further. But I just want to kind of have a broader session around class of ideas. So I don't know who wants to share the FRDPARRC. And people can also just, since we don't have the screen working, we still don't have the screen working.
I can pull it up. Would it be easier for
all
me to
just
just
join
put it
on
on and
Zoom
let
and
others talk over it? Yeah, okay. Sorry, let me just do that. Okay, so I'm just going to share the screen again, but then I'll let the rest of the team that put all of this together chime in on the different aspects of it. Can people see this? And we can also share a link, I believe, even directly in the chat, if that's helpful.
Yeah, and then also just Discord project. At this point, everybody should have a project Discord channel. So up till now, we've been doing everything, but now every team should have a dedicated channel.
Okay, so that link should now be shareable to everyone in the chat as well. Yeah, should I keep going? And does some of the others want to jump in here?
So who wants to just walk through FRDPARRC? What's the most interesting, exciting, something that really resonates with the team amongst the sets of functional requirements that you might think about tackling this problem? Where might you want kind of the focus for everybody for the brainstorming should be?
Yeah, Manu?
Yeah, go ahead.
Yeah. So what we were thinking about is like Dave said, it's like a multifaceted problem. So we wrote the FRDPARRC in such a way that we could tackle the issue before it's processed, like the cassava in the soil, and then during the processing of the cassava, then after that in the human body after it's consumed, like detoxification and something on the lines of that.
Yeah.
So for the first thing, some of the basic things that we thought of were like
yeah. So just for now, I just want to make a comment on that. I think everybody can see that, right? I mean, this is a really beautiful organization because in some sense you walk through the source of the problem all the way to every step, and there might be a solution at any one of the steps if you can break the chain. And then all the way to the end is like, what do you actually do with people who have the disease at this point? And I mean, I love this organizational because it has clarity in where you might focus, and you can then feel like, although we're focusing on one layer, you can always jump back to the other layer. Yeah, go ahead, Mrinal.
Yeah, okay, yeah, so when it comes to the cassava as such, when it comes to the plant, the main cassava that's consumed in Congo and the rest of Africa is actually the bitter cassava, and the bitter cassava is what contains more of the cyanogenic glycoside, and it's called linamarin, and that's the predominant glycoside. So the reason the people in Congo and other parts of Africa use this bitter cassava is because it's more resistant to the climate change and things like that. So given the climatic, given the issues like global warming and things like that, changing precipitation and sunlight, bitter cassava is most likely what they're gonna use. So when it comes to replacing with sweet cassava, the problem is that it might not be as resistant like as the bitter cassava. So there has been studies where they tried to, there are these cytochrome genes that are found in the mitochondria of the plants that produce these toxins, but they try to knock those genes off and made a hybrid, like a recombinant variety of cassava. But the problem is they don't have as much yield as the normal cassava varieties. So that was especially not practical. So the other thing we thought about was that, is there something in the cassava actually produces linamarin for two purposes. One, the linamarin is produced as a source of nitrogen to make essential amino acids like aspartate and glutamate, and also it also acts like it also acts as like an antioxidant in the plant. So we're thinking like if there are any microbes that coexist with cassava that would help in fixing nitrogen in the plant, or somehow it could maybe reduce it. Somehow it can influence the production of the linamarin in the plant cells, because cassava is actually from South America. It's not from it's not from Asia or Asia or Africa. It was actually introduced by the Portuguese to to Africa. So the populations living there are actually not used to eating cassava as such. So the people in South America have a
Just Mrinal, just thinking about now the plant itself, and then in general the kinds of interventions around plants. Is there a criteria that in the cassava, the portion of the plant that's eaten is exactly where these sets of compounds are concentrating?
Yeah. So
is there a reason?
Yeah. So I think like evolutionarily, the thing is that like I said, it's produced in the leaves, and the plants transport it to the roots. That's where they store it. I think it's around 84% of the cassava roots made of carbohydrates and 8% fiber, and the rest of it like 1% fat and things like that. So this linamarin, those cyanogenic glycosides that are produced in the leaves, that are transported into the roots. And the reason plants evolved to make this is actually it acts as a during the droughts and during like low yield during the summer, it actually helps them have a protection against herbivores, and at the same time they also help in as a source of nitrogen to make the amino acids. And like I said, during this, it acts as an antioxidant during the heavy sunlight causes reactive oxygen species production in the leaves, and it helps to the antioxidant effect of the linamarin.
I see. So there is a biological function in the plant.
Yeah.
It's not both the leaves and the roots are consumed as well.
I see. Yeah, I mean, I think one thread to think a little bit about is amongst the breadth of places where you can attack this. I think as a team, you guys should be thinking about every one of them. Which ones do you all feel is the strongest and the most implementable? Because I think Yuki had mentioned something earlier on, discussions around processing. I mean, there is clearly one approach of, a new genetically modified cassava that is both resistant and doesn't have an approach. And that, I mean, I could see that approach would be very valuable, but at that same time would take a much longer time. While I had heard you guys being excited about processing, so the battery is about to run out. So I'm just curious, because time is short, amongst the many criteria.
Oh, I think we lost you, Manu.
I think we lost the entire laptop.
Yeah, maybe the battery died. Okay, so why don't we use this as an opportunity amongst here? I think what Manu is leading into is saying that we should focus on let's just focus the rest of the discussion on processing, because I think that's a more tractable thing for people to get around. Yeah, so
for sure.
So
and I think we should also focus on a little bit in terms of like the setting, right? Like there's definitely the centralized processing that's seemingly to get rid of most of the problem, but then what about the more practical one where they're the water constraint as well as the acidic constraint?
Perfect. Okay, so let's try to get that across when they come back on and just say
we're online again.
You're on. Okay.
Yes.
So just quickly, what we were saying is that we think focusing on processing makes sense to lead this discussion. There are definitely industrial processing methods that effectively detoxify cyanide from cassava, but we are talking in these affected regions about very, very low resource environments where water is often the limiting factor. And so the general processing workflow for cassava involves peeling the roots, soaking the roots for a long period of time, which releases some of the cyanide from the cassava, and then drying out afterwards, pounding it into flour and using it to make food products after that. One of the major challenges with detoxification in this way in these low resource, low water environments is that there's not enough water to be able to let the cassava soak for long enough, or to leach out the cyanide for long enough. And then connected to that, also without a lot of water, it ends up being acidified, the water that they're being soaked in, and this affects the enzymatic properties that are able to be important in that detoxification process. So
And
that's kind of
Can
processing.
you put some timescales to it? So the ideal case scenario would be peeling and soaking in how long and how much water, just to see what has historically worked. I mean, are we talking about, I'm curious about the timescale. We don't need it.
Yeah,
anymore.
Oh,
We don't, because we have it now.
okay.
Yeah, we can just shut that. Sorry, Dave, can you just say the amount of time used in soaking?
Yeah, I think Murnau or Meghna would be better at describing it.
Yeah,
Go ahead.
you should, historically, they used to soak it for 3 to 5 days. But recently, because of the drought and all the conflicts going on in the area, and there is a commercialization of the whole process, and the things that they get to buy is often not processed according to the rules. That is, it's soaked for lesser time, maybe 1 or 2 days. So that's the thing that's increasing the concentration of the cyanogenic glucosides. There is one thing else I would like to add to the point if this cassava processing. That's boiling can actually reduce the concentration of the cyanogenic glucosides in cassava. But this is not part of their cooking process. The practice that has been, the cooking style that they follow in Congo and other African countries is without this boiling component. But if you can add this boiling element to the cooking process, maybe that can decrease.
Yeah, so Meghna, you said
Yeah.
something very important that can totally take many different directions. Do you guys know by the numbers, what percentage of cassava consumed is brought from the market? Because, I've seen many places I go, I see cassava processing just happening around. I mean, this is the most common image that you see in a rural context in Africa, people pounding, but then times are changing. So are we talking about 50% cassava that's brought in is processed industrially versus 50% still done at home, or what might the ratio be?
I think in
Where would
these,
we
at least the regions I visited there, none of it, I would assume, would be industrialized at
Got it.
all.
Okay.
Yeah.
Okay, so it's still worth focusing on the home processing as the primary criteria.
Yeah, and that's not to say that it's
Just for
not
time
sold
being.
in flour at the market, but this would be very small scale. This would be,
Yeah.
people with an individual little patch in their
Oh,
yard
I see your point. So although it's being sold in the market, it was still just processed by somebody at home at a slightly larger. Okay, that makes sense.
That's what I would assume, yeah.
Okay, so it still is relevant that we focus on an individual or non-industrialized processes.
Absolutely, yes. Yeah, it needs to be very affordable, and it needs to be very, very small scale and very low tech, let's say.
Yeah.
So it seems like, can you do crop replacement? I mean, it seems like a better thing than allowing this toxic thing to enter the food supply to begin with.
I think that's a tough one. There can be lots of people that can be thinking about, but as you know, Congo, there are so many other sets of challenges that is easier said than done. I think it's still worthwhile because social practices around food consumption are very strong. It's very, very difficult to just really flip that around. I think
but Mrinal said this was brought from South America, so I was just thinking, since it's not native, maybe
many hundreds of years ago. I think now it's part and parcel of life. I mean, I think Dave can comment on it. He just got back from Congo, so.
Yeah, I can also say that this, this crop is a blessing and a curse. It is incredibly hardy. It's incredibly stable. And that's why it's consumed by so many people, and it meets caloric needs for so many people because of how hardy it is. But so I think that would be a real challenge to displace that, because it's just so solidly in the culture and the food, and it's such an accessible source of calories. And for the most part, it is not a problem. It's a problem, it seems to be, when the stars align and either inappropriate processing aligns perhaps with other factors. But surely effective processing of cassava, we understand or we believe, would solve this problem.
Yeah. So Dave, a quick question around, I think the class of ideas that I'm thinking about is what sets this time scale of soaking, and that comes from diffusion. You have certain things and there is a diffusive time scale. So if you guys post some pictures, I'm assuming the cassava root is all hard and very solid. You soak it in water, it starts to soften in some way that allows the diffusion for these sets of things. What if you increase the surface area of something like that massively?
Yeah.
Could you reduce the amount of time, and could you, because that interface would change, and in the end, it shouldn't matter because you're gonna pound it, dry and pound it anyway. So the form factor of the root doesn't matter so much. So is there a context when they are putting it in soaking? Are they just literally throwing the roots, or are they chopped, broken, shredded in some sense?
Yeah, I'll try to find could somebody find the video that you found previously, and maybe we could show that. But
Yeah, let's just watch that video. I
The
forget
root is very hard to begin
Yeah.
with, and it softens up by soaking it. So, and
then
yes, there's
Yuki, I don't know if
diffusion,
you
and
are
there's also
back.
enzymatic.
If you are back from your conference, I would love to eat some cassava next lab meet. Are you back? I'm actually
No,
Oh, you're
I
not
would
back.
love to, but I'm actually on an academic exchange in
Oh my
Germany. I won't be
goodness.
back until the
Okay.
course is over.
Who's cooked cassava here? All right, Henry, are you willing to bring it Friday? Either processed or just the raw, like whatever form of cassava people can bring into the room. I think this is what was very important about the ostomy bags. For your project, that's the first thing that you should all do is get physical with whatever you care about. Because I mean, what I'm hearing is it's a classic diffusion problem. And we understand quite a lot about limits of diffusion, and clearly some amount of water is needed, but how do we think about reducing that time? Okay, I think I see a video is about to play. So
Okay, yeah. So okay, let me find the right spot.
and then can you guys be posting all of these videos in the channel?
Yeah.
The dedicated project channel.
Okay, so this is an older video here. I don't know if the sound will come through, but what we're going to see in a moment is the pits where the cassava is being soaked here, right? Okay, so here's somebody harvesting the roots of the cassava, peeling the outside, and you can see it's very hard, and then they're soaking it in these pits here that are where the cassava is sitting in there for a number of days. And then after soaking, they pull it out, dry it.
Yeah, I just saw that I
And then
now
pound it,
see where shredding it into tiny pieces is a little bit tricky because it was literally in a dirt pit.
Yeah.
And so you want to be able to pick it up easily, but you can also imagine a simple way that your shreds are still shreds, but you can pick them back up.
Yeah,
Like
and then I think
using an old mosquito net as your netting that you throw this in.
right. And
Also,
and
like
then
one
this
of the
is processing
important-
afterwards. Sorry. All right, go ahead. I'll stop talking.
No, the other important point that we want to bring out is that this is the pit that the whole village is soaking over and over and over again. So you can imagine the concentration of cyanide in that pit.
Yeah, I guess because the water itself is a very limited resource, and you wouldn't use drinking water or other things for it, because that's a very precious source itself too.
Yeah, and one part of the chemistry is also it's an enzymatic process that's happening there, and those enzymes do not function at low pH, and the water ends up getting acidified, which further contributes to this vicious cycle.
Yeah. So can you say a word or two more about the enzymatic process? So if you were to think about, I'm thinking of alternative ways around that. So the enzymatic process is that it's not just a diffusion of one compound out. In the water, something starts to change when the root is exposed to water and peeled off in this way. I mean, is there an osmotic pressure or an osmotic drive here effectively?
Yeah, Manu?
Yes.
Yeah, so the usual method that we use to process, the basic idea is that the linamar is contained inside the cell of the cells that store the food in cassava. But while there's an enzyme called linamarase, and the linamarase actually sits in the cell wall of the cassava. So if you I'm sorry if I'm drawing it wrong. But
No, no, keep going, keep going. We're not seeing anything you're drawing because we're watching the video.
yeah, you see this
Oh.
like this is the glucose, and then at this first carbon there is a carbon attached to it, and to it there is a cyanide group attached. So the thing is that this is the bond that's broken, and that's how the cyanide is released. So the linamers breaks that bond and releases the cyanide. So what they essentially do is they grind the cassava. When they grind the cassava, the cell walls break, the linamers and the linamerin join in, and the cyanide is released. It becomes something called acetocyanohydrin, and then it breaks down in this
And I'll
acetone.
just to understand this, is the enzymatic process happening at cellular scale at the point of pounding? Because cell lysis is happening at soaking or at pounding?
Yeah, in soaking, the bacteria actually causes the process to happen, and it breaks it down, and the cyanide is released. So
I see. So
I think Dave was saying.
Yeah, I believe I believe it is happening in the pit. So I think there's a lot of linamers in the skin, and peeling kind of kickstarts this enzymatic process that then continues in the wet environment. Whether there is still enzymatic properties left after it's already pounded, it's possible that there was a public health intervention to do an additional wetting step of the flour after it's pounded that results in additional release of cyanide, whether that's from an enzymatic process or just
really
from
I see.
So that's been the major public health intervention, and if we can think of more things that are more like on that level but more effective, that could be a good point.
So here is what I'm thinking about. I've been telling this to all the teams, is to find gold standards. Like for you guys, you have a gold standard. You can actually put an ostomy bag and feel it and say, am I better than this? So in this process, is there a very clean assay? If we got cassava in everybody's hands in the team and anybody who wants to help, is there a very clean assay that we could watch the amount of cyanide go down and mess around with this process? Is there I mean, I saw that little litmus paper just for a flash of a second in the video. So seems like there is some quantitative assay that can be done at any time of the product and draw a red line to say, this is consumable and this is, I'm not saying that that intervention will work because people need to eat, but just it acts as a gold standard for us to test ideas against something.
So I have one question like this. How do you incentivize this? Like if I'm a bad actor here, can I just put my cassava in, let's say I'm growing cassava and I can just put it into the market without even doing this cleanup? So what makes people do the process properly?
Apparently people can taste it. I've been told you see people in the market tasting the flour for bitterness. So a bad actor can't necessarily, I don't know how much that plays into it, but people are aware of this. And this is a poster about the wetting method, that public health intervention.
Okay.
And one more quick question. What if you were to split the thing, not like make small pieces, but just split it into 4 or something lengthwise, so that you allow more access to the internals, but it's still pickable from the mess of the pit.
Yeah, I think that's just the same direction of just diffusion. Diffusion should be one idea. There's a comment, Jess. Yes.
Oh.
Did I get your name right, Jess?
Oh, Jenny.
Jenny. I thought I was so close. Sorry. Yes.
Yeah, I was looking into like there are a few I guess bacteria and fungi that could break down cyanide that are used in industrial processing, and they're mostly soil fungi or soil bacteria, right? So if you introduce that into the pit,
can you write that?
Yeah.
Just bacteria and fungus breakdown.
There's a fusarium and there's also pseudomonas.
And again, there's probably plenty of bacteria. I mean, I think one way that I'm looking at this and watching those videos, frankly, I would venture out to say we don't really understand what is happening, everything that's happening during this process from a soaking in an open pit kind of a scenario, which is what makes it also exciting. There might be many, many other threads that could be thought about. And focusing as a team also on the understanding of this process is a very valuable way to make a breakthrough in some sense. Yeah. Yes,
I
Tanvi.
guess building off that, I was just curious about like the microbiota of that pit and like
Mhm.
what.
Yeah, so can you just write microbiota? And you know, the not just the pit, but the plant itself.
Yeah. Because there has to be things that are
Yeah,
going in.
I mean, it's a giant pit of mud, so you can already imagine
Yeah.
millions of things in there.
Yeah.
Yeah, go ahead. Somebody online, Dave or
Yeah, Manu,
yeah, go ahead.
there are these bacteria that produce an enzyme called nitrulase, and that's something that can break down the cyanide.
Yeah,
Yeah.
that nitrulase. And then I just want to mention something from Madagascar, something that I learned. It's only partially related, but it's relevant to look for sources of ideas. So bamboo lemurs eat a ton of bamboo, but bamboo has an incredible amount of arsenic. But as an organism, it's they have figured out the microbiome to process that arsenic, and then they find that in a niche. And so, you know, thinking a little bit about cyanide and thinking about other sets, this space of the microbiome, it's very interesting, and it would be useful to think about if there is an organism that has evolved to eat these roots, and still, does any animal eat these leaves? And what might be the gut biochemistry there, just to get inspiration even.
Yeah.
Yep. Since we're talking about the direction of microbiome and a more bacterial approach, I also just want to mention that there is also the chemical approach that potentially we could think about, because the water pH becomes so acidic, and that sort of inhibits enzymatic activity. So potentially, if we could also think about the direction where we can calibrate and bring it back to a more basic solution, that might also be a good way. And yeah, so that's also something that
that is very easy to do. Like I can imagine an ultra cheap way, baking soda style, to be changing and thinking about the overall pH. And you know, yeah, I think are there other chemical approaches that come to mind other than just modulating pH?
We talked about catalysis or something, some way to more efficiently, you know, if water is the limiting factor and your water is becoming toxic that you're using, is there some catalysis? Is there something that you could be, you know, filtering your water through, for lack of a better word, that would get rid of the cyanide, but then allow you to use it for more soaking to more effectively extract?
Oh,
so
that would be, yeah, because you know, in the end, the diffusion is that there is a gradient. The more cyanide you collect, the less then it's going to be pulled out.
Sure. Yeah.
Yeah, so something that cyanide binds to very, very well, for example. Okay, I think, yeah, we can spend a whole day on this. So we're going to take a pause on this. But I mean, this is exactly why, I mean, what an incredible problem. And also at that same time, it feels very, very tangible. But to me, it's the necessity for this to be just literally done by somebody in their house really puts the kind of constraints that, although we can imagine, and I think we want to broaden this, but there is a very harsh reality which was visible in those videos of just, you know, immediately that, yes, this would be a very nice research project, but the reality on the ground is it has to be a practical process. So I think, I mean, this is why this is fantastic that you pulled in some of the videos, and Dave, you've actually seen this process in real life, because eventually broaden this out massively, but apply that filter very rigorously. So then you say that it is something that must be done in those sets of conditions. And again, you know, just the point that you said about stability, the fact that it might not be water, but people are moving all the time. I mean, many of the migrant populations, especially in instabilities, that could completely change how long because this is food, so you don't want to leave it behind. And if you get into that situation, this shortening of the time itself is quite valuable. If there could be a process that could be done in a day,
right,
you really are talking about much more sort of an improvement in the context of if people really do have to migrate and move. Okay, what time is it? I think we have time for one more team. Is there team members here in one team that's complete right now? You guys have everybody in your team? No? Who
Maybe
else?
I can.
Oh, we have Shujo and Tanvi,
Yeah.
right? So why don't we do you guys as the next team? Before we start on that, I just want to make sure everybody does have a project, right? Which ones are you debating on? Just so that, let me just cover.
I think I've decided.
All right, when did that happen? Just right now? Okay, and which,
That one is gonna be the open.
okay, and then you've spoken to the person who else is in
Yeah,
the
I
team?
text them and we discuss.
Okay, that's perfect. And then do you have a project yet?
Yeah, I'm deciding between two.
Which ones?
Using solar power to like convert water into like usable water,
Uh-huh.
and then something with like converting methane into biogas or like
Uh-huh.
collecting waste.
And then do you know who is in the team? Who
Yeah,
else?
one of them we've been like talking on Discord.
Yeah, is that someone online?
I don't think he's on.
They're not online. Okay, yeah, so I think one thread that it's, to take this class, you must do a project, like that's not negotiable. And the sooner you make that decision, it's about jumping in, it's not about, we often, the process is where the fun is. Frankly, it doesn't matter which project you actually choose. The purpose of the class is to make sure you learn the process. And then, I mean, you can already see just how rich every one of these problems is. It doesn't matter what you choose. Once you've gone through this process, you'll apply this process over and over again. So please, please, please make sure that you're committed to a project at this time, because we will not have enough time otherwise. And you won't really go through the, it's not participatory. It's something when you feel a little bit of ownership. Okay, so Shujo, Tanvi, you guys can take next. I don't know if you want to open up a FRDPARRC.
Yes, I'm gonna share my FRDPARRC. So.
And then again, we'll do this rapidly. So choose, after the broad picture, choose one specific thing that we might dive in.
Okay. So our team is interested in the quantifying the number of the invasive species in aquatic environment. So, and we specifically focusing on the red swamp crayfish, which is red color crayfish, which is invasive species in many European countries as well as Asian countries. And so we are thinking to
also just as a practical matter, are there also waters here that it's invasive in the US?
It's in Jasper Ridge.
Awesome.
I've caught
Okay.
crayfish from Jasper
Yeah.
Ridge.
Can you bring it next Friday?
The crayfish?
Yeah.
No, but
You
we
can't
can
catch
go
it.
there and test it. I can ask, but they
Yeah.
don't care as much about collecting it because it's invasive.
So you can collect it.
I can collect it.
Yeah.
It'll just be a little bit of a process, but I can
Yeah,
figure
but
it out. Yeah.
write the
Yeah.
paperwork already for it.
Yeah.
I think this is what I'm trying to say. It's very important to hold the problem in your hand.
Yeah.
So Shujo, actually go out, whichever city you are in, find the closest crayfish. It's very
Yes.
important to kind of hold it in your hand and say, this is what I'm trying to tackle. Okay,
Yes.
go ahead.
And so basically, so this FRDPARRC is I wrote myself, so it's not our team's consensus, but I personally thinking about the imaging-based detections. So basically using the flow-through microscope and also the imaging-based classifications, and so which is basically inspired by the planktoscope approach. And also, if possible, I also think to sort the crayfish for the separations. So here, basically, so first our goal is how to monitor it and how to detect. So for the monitoring, I use, so as I said earlier, I use a flow-through microscope, and then classify the larvae of the crayfish using the imaging-based classifications.
Yeah, and then Shujo, can you say a few words about
Yes.
just the constraints on the size of the larvae, how abundant
Yes.
they are, and then while, can we get another note taker? It is kind of a fun exercise for anybody who wants to pick up the pen. Yeah. Yeah, and then you can erase it. We have photos of that. Yeah, yeah, you can erase the whole thing. Okay, go ahead, Shujo. Yeah.
Yeah, thanks. So basically the larvae or the eggs is basically 1 millimeter or 500 micron in diameter. And the abundance is, so I briefly quantify the numbers from the literature. So basically the for the density wise, it's approximately 10 in every square meters. So that's maybe quantitative information for the abundancy.
After they've invaded or as they're invading, because part of the detection issue is like,
Will change as a function of time.
yeah, change the function of time and like part of the detection.
Yeah,
is
so you
like-
can think a little bit about a detection limit.
Yeah.
You could say, oh, we want a detection limit that's 1 order of magnitude lower than when they have established themselves, which is say 1 larvae per square meter. And then I'm assuming, do these larvae have some behavioral preference? In the larval stage, are they in the bulk of the water? Do they prefer benthic? Where are they finding their food? So if you were to sample the water, when you go to a stream, would you sample on the surface? Would you sample at the bottom?
I think the larvae or egg is basically on the bottom of the lake.
Just so that we have like a full working space. Yeah. So that's kind of, this can be as part of the constraints and by the numbers, which will be mapping the behavioral, where would you sample it, what the numbers are around the detection limits, and effectively, is there a preference that they have to running water, or is there something that we know about crayfish larvae from a behavioral point of view?
A lot of the existing, like I was looking at the existing EDNA filters, and a lot of them focus on like surface monitoring.
Surface. Okay.
Because like, the dispersal mechanism is surface, and crayfish is like on the bottom because they're bottom.
Yeah, but the larvae disperse on the
will
surface.
will primarily disperse.
Yeah.
to the
Are they
surface.
swimming larvae? Or?
I'm not sure.
Yeah,
I think they are,
they
but
must.
I would not like
Yeah.
bet on that.
Okay, and then I think one thread to think a little bit about on this would be the samples can be collected. They don't have to be processed in the field because the larvae, do they have a cuticle? The larvae themselves, I'm just thinking a little bit about, is it a soft-bodied larva? Can it be fixed? Because you could imagine a surveillance system that a massive number of people collect the water. I don't know if you guys know this, there is this organization called Adventure Scientists. It's an NGO that started some time ago. And if you ever go hiking and you have a water bottle with you, you can collect some water and then you can ship it to them. And there are different people that propose projects, while there are people who just love being in glaciers and very high mountaintops. And then many people are not going to those locations. So there is a kind of they act as a middleman type of a ground where I could imagine that the processing doesn't have to be done real time. And then in terms of Shujo, the lifetime, what is the larval lifetime just overall?
Larval lifetime.
Like how long is the larva remains a larva?
That's, I really don't know about it.
Yeah.
But
Does anyone know?
Push back. I think the processing does need to be real time. I think like in terms of like actually detecting like invasions of crayfish in like a meaningful way, you need to be able to, like the issue with invasive species, you need to be able to detect this on like a week-long time scale in order to put in place sufficient mitigation activities.
That's also related to
Yeah.
thinking a little bit about the larval lifetime, for example.
Yeah.
And I think that's a very important thing.
Yeah.
So let's
Yeah.
write that down as a constraint, which is what is real time in the end? And how real time does this monitoring need to be? And we know for different sets of problems, that number has to be quantified somehow. Yes.
I have a question. So I'm reading about this like a bit online, and it seems like the eggs are just attached to the mother until they're basically like actually hatched. And
Yeah,
at
but
that
then
point-
they become larvae.
And then there's like a few sources say like the larvae also just like version of like
Mhm.
eggs. So would that actually like impact if you're using like the scope to detect?
Yeah, my intuition is that once they're in the water column, they're being dispersed physically, and that is the mechanism for them to go to new areas. And so there is some point of time that the larvae are in the water body itself. And I think it's this is why I was saying understanding that behavior becomes important. And for dispersal and invasive species, understanding the behavior is critical. Like, for example, I mean, one way of thinking a little bit about this is mosquitoes. We really care a lot about mosquito and mosquito dispersal because of diseases. But I'm curious if any of you know, how far does a mosquito fly? So if you had a mosquito, if you know the answer, then you shouldn't tell. But I'm just curious if you guys can guess. If there was a mosquito in its entire life, do you think it's a flying insect? How far do you think it'll just fly away? It's a tough, but I just wanted you to build an intuition for it around invasion. Like hundreds of kilometers, 1 kilometer, 10 meters, 1 kilometer. Yeah, hundreds of kilometers. So it's actually a very small perimeter. So it's, much less than kilometer, even hundreds of meters, which is very interesting. But then there are other means of dispersal that people discovered after a long while, that mosquitoes are taking the thermals, going to very high altitudes, thousands of meters away, and riding the currents in the atmosphere and then landing in totally different countries. And so it completely shattered our understanding, because Mali, you were getting all of these mosquitoes that in the dry area cannot survive. And then suddenly somebody discovered that they were like para-trooping in. And so these types of invasive things this is why I'm saying the biology of this is very important to understand, which is what mechanisms these larvae are taking. Because a little bit what might be very interesting as you get into the biology of these larvae is not only that it might become an approach to intercept them for monitoring, it might also be an interesting strategy for thinking about control. Yeah. Like if what you said is true around surface water, that is by itself a very interesting approach to think about. And then in terms of like some amount of cost analysis and other things to think about, how urgent are crayfish in people's minds? Is it something, who does the monitoring currently? Like, are there citizens that care about it, or is there a body, like a government body, that is charged to engage in this?
There's like just something, I guess, I mean, I'm not sure about the situation in Japan, from what I've gathered in the US, like most national, and Singapore, most national park services are engaged in monitoring invasive species to some extent. I think iNaturalist to some extent has been supplemental for like finding these things, but mostly it's a different domain of the National
But
Park
what
Service.
falls in its National Park Services, for example, would be so if you're designing a product, they would be the user effectively.
But
Right?
they're like the EPA also does like some extent of invasive monitoring. I think it depends on exactly what kind of dimension is being put into.
Yeah.
I think.
Yeah, I think the threat to think a little bit about who's boots on the ground, because whatever we make is the boots. Is the solution designed for citizens? Then we just focus on citizens. Is the solution designed for a national park ranger? And it might be that there are other people, once the data comes, that they process, but they are not interacting physically with the solution that you all must be thinking about. So I think narrowing that down would be valuable, because it'll give you an approach to say, oh, how much time are they really spending in this ecology? If it's a national ranger that lives right next to the stream, you can really imagine a very intense monitoring in a particular. Oh, also seasonality. Is there a rhythm to their breeding sites? So just kind of annual rhythms. Do you know, Shujo, is there a mating
I
season?
think it's basically autumn to spring
Okay.
for the laying eggs.
I mean, I'd envision a tool that's like accessible to like people who are monitoring or who are managing like water bodies. So like which I think can apply to like anywhere from like a biological reserve to a national park, but it's like people who are in charge of a watershed and in charge of the biological diversity of the watershed.
Yeah, so try finding an example. And since you said Jasper Ridge has it, we have a project that we're just starting in Jasper Ridge to map the entire microbial biome of the aquatic.
In Searsville or in the
in
Ridge?
Searsville,
Okay.
yeah.
So
Yeah.
sort of a
So-
last monitoring.
100 years ago, what was done, we're redoing the entire thing with a completely new scale and technology. So Searsville is this water body. It's amazing historically and stuff. And so there's a PhD student in my lab. And I think this will interact very well, Shujo, because we are now going there very regularly. We're starting to write our permits now, but the idea is to every 2 weeks collect water samples. And since you said there is crayfish, I'm super excited to see whether we will find and see larvae. And I think this is something that you should also just try to do because very quickly having some class of samples, it might be that you can't find crayfish. You can go to some other, for example, we are also working with a couple of oyster farms. So we have oyster larvae. We have a couple of different types of larvae. You can also think a little bit about, for the ML side of the story, there is value, and we are currently writing the ML pipeline for oyster larvae, and we can give you guys the data, and this will just get your hands wet in thinking about how hard it is to write an automated system that will pick every time it sees a crayfish larva. So I think it's a very tangible, immediate thing that you guys can do, because with the oyster larva, we already have the data and we have the pipeline, and then you guys can improve it.
I know she was looking for like image databases of carfish that I was struggling a little bit of finding, I think.
Yeah.
Specific images, so.
Yeah.
Which
Yeah.
is-
And I think the larval data sets I've found for a lot of things, we just have had to create them. But it's, yeah, I think now having seen what OpenAI can do with synthetic imagery, maybe you can just generate larval image. I don't know if you guys saw the videos yesterday. It's insane, the text to video. We're going to be living in a fake world with our Apple Vision Pro and completely regenerated reality. So yeah, I think it's a little bit hard. It's perfectly okay to choose a data set for like copepods is an easy one. I can give you guys the Ecotaxa copepod database, which is 2 million images, and it'll just get your kind of process going in terms of, given a data set, how do I train a model, which is, a couple of days of work, but it's pretty straightforward at this point. Okay, any other comments on crayfish? Anybody has ideas around threads? Any other words that should be thrown in there? I think clearly plankton nets is something that comes to mind. We've been working on a lot of low-cost plankton nets that people can make at home, because then you get to filter stuff directly. So just write the word plankton nets, because beyond the scope itself, the nets need to be available, or how you sample and filter water.
Yeah. I guess I'm curious, like in terms of like what I was talking about earlier, like high throughput filtration systems for like an EDNA assay as well as like
and I think so. Then it becomes super exciting, because then break this problem down into 2 arms. One is microscopy and the other is DNA. And you can tackle on the eDNA side, while Shuja, who else is in the team? Is there anybody else?
We have another two
more people. Yeah, I think it's perfectly okay in the team that you can be excited about a problem. But if you really feel strongly about also a second approach, that you take more of a burden on something, and it's still working towards the same problem, but you're dividing and conquering in some sense. So Tanvi, I'll give you the paper on the filter paper side of that story for what was done for single viromes in gels. And we can start from there. We have that entire protocol. We use that protocol. I think to a certain extent, the question again goes back to doing some calculations to say how much water do you want to filter? And then a general problem that I'm excited about is really a better way to filter water because of just, that's a much broader problem. We can use this as an excuse. And currently the situation is so bad that when we go to sea, we use these, have you guys seen in construction, there is this type of a tube that's used for putting glue, and it has this little pump that you cups. Yeah, so we basically use them to filter water because you need enough pressure to squeeze through these very fine filters because again, we're actually filtering viruses. And so the pore size is 100 nanometer or smaller, and it is so painful. It's just, you will sit there, if you have a liter of water, that's all you do. And again, you just keep, so I think.
And it's like super painful when it's like the road is rocking, if you're like in a
it's
completely unfeasible in field conditions, but it's also like, it's the only thing you need to do in the field.
Yeah, which is terrible because, you know, you want to think and effectively. So anyway, I think just purely water, because all the rest of the pipeline is actually, so this is a sub problem. Like we started with crayfish, we focus on eDNA, we realize what the problem, because everybody else has actually solved. It's very valuable to crack a meaningful problem. So I think it doesn't matter how deep you go, because then you can build a solution out of it. But I think the problems that you choose really have to be real problems. And so I think on that side, it might be that then that solution ends up being useful for other things. Kendrick, yeah.
I was just wondering, is it possible to make that electromechanical?
Yeah, you can think about motors and others. So I think from a battery point of view, that's very useful. There's also, you can think of the gearing. You can gear up a low power motor to really get the kinds of pressure that you're interested in. Some of the other aspects of that is ergonomics, you know, using gravity. There is a vacuum pump that one of the students, I think Samir, who was 2 years ago, we built an ultra low cost vacuum pump for electron microscopes. So, I mean, just so that if you guys are feeling that, oh, you're being ambitious, 2 years ago, we built an electron microscope as a class project, and it actually works. And so now we're building a large team. So it's just, I mean, it is quite remarkable what you can do if just the right kind of people come together. The thing that I'm always reminded on vacuum is, I don't know how many of you used it. We used to have these kerosene stoves as a kid, and there's a little manual pump in it that, I don't know, I think in some campings you still use it, and that pressure is used to drive the fuel. I think the folks in India know probably what I'm talking about. So anyway, there's a couple of products that are out there that have small pumps that are integrated that you guys can look at in terms of its efficiency itself. But it would, and I'll send you, Tanvi, the data sets on what we did on the vacuum. I think it was not designed for this, because here you're pumping water, there we were really making true vacuum.
Yeah, but like even like a lower like version of that, it maintains a constant pressure.
Yeah, no, it's just the class of ideas. You know, the place to start is some of the water pistols have gotten pretty efficient. Like how far you can shoot in these toys, and you can really look at what is the efficiency of something like that, how much pressure can you actually generate. Okay, any last minute comments on anybody online on this problem? We're gonna stop at this point and then kind of pick up the FRDPARRC. But I think what I want to do is, for the next entire week, this is all that we'll be doing for every team, to just make sure that we have done a collective session for all teams. So just be prepared when you come in, because you can see it's very valuable for everybody else. This is the only time everybody will be focused on your project. So just when you're presenting it, be super prepared on, you should have done something like this yourself. So then you say, this is what we've already thought about. Let's expand beyond that. And then we'll pick that. Hope, maybe one thing we could do is we could do a sign up sheet. So that way we keep it to 15 minutes. And then let's see on Tuesday, how many total teams do we actually have? Do we know finally?
So I have 1, 2, 3, 4, 5, 6, 7 teams, and then I'm not sure how many solo teams. So there's 7 that I know.
That everybody here is in one of those 7 teams. I think I heard about 2
No,
separate
not-
projects that are not. Have you guys put the entry in the, there is a master list you have, right, for project?
And so I can share that.
Yeah,
with
can you just remind everybody to make sure that, please make sure that you guys put the project in the master list. And then thanks again, everybody connecting. And one of the threads that we'll try to do is, I am still happy to leave open times, late evenings. I think last Friday, nobody signed up. So maybe then I realized that that might not be a good time for international teams or people here. I am happy to do Zoom calls around 9 and 10 p.m. Many of the days, because that's when I get free, finally at the end of the day. And if that time is useful for teams in India, so Mrinal and Meghna, the Ethiopia chat, let's definitely move that to say, maybe Tuesday, 9 or 10 p.m. would actually work. Monday, I have to be at a meeting. But just if you want to continue these types of brainstorming sessions, that's just a time slot that I can always make available to people. And I don't know if it works for other people though, because now there are so many other time zones. But just, if you just let me know one day in advance, I can make that available. This is also true for just here. It is a late time and I don't expect it works for everybody, but it's just, it's useful if you guys want to take that time slot. Okay, any last minute questions? Are people feeling comfortable about what you need to do? So at this point, make a list on materials if it's very obvious. I don't know if you have all gone to the hardware store yet. What do you really need? Hope is going to be maintaining the list, and then based on demand, then I'll decide kind of budgets. It's fairly straightforward. You know, it's I talked to Ross. Mong is going to give us more bins that you guys can store stuff here, so that's all ready to go. So bring materials in. I think from now onwards to the rest of the end of the session, it's much more about actually doing testing, experiments, spending time in the lab rather than lectures. Because I think, you know, we won't make any progress otherwise. I mean, this is helpful, but only so helpful. Like, I mean, this generates a lot of ideas. Now you have to cross them all out. How many of them don't actually work? So, and then if somebody can get some cassava, Henry's on the hook for cassava next week. Yeah, and then can you get raw and powdered and something cooked?
Yeah, I don't know how like good it would taste without the other aspects, like that you eat it with. So I can make it.
We can play with it.
Yeah.
You don't have to eat it.
Yeah.
I want to taste it too. I've had it many times, but I think it's kind of fun to have a visceral object to be thinking about. Yeah. And then, you know, you don't compel to do it. If you feel that that would be a joy for you to just do, then do it. But otherwise, just bring the raw materials. Like, oh, this is a cassava powder. Because everybody should think a little bit about what you could do. So anyway, yeah, and I think Yuki, you can have something wherever you are and share that online if you can find it.
We'll try.
Okay, bye everyone. And I'm assuming, I mean, I think we had a couple of people already in Africa. In this current group, I don't see anybody connecting from Africa, right? Mrinal and Meghna, you guys are not in Ethiopia, are you?
No, we came back.
Okay, yeah, you had your share of cassava.
Yeah, we eat cassava here at our home. Yeah.
What?
Yeah, okay, we can consume a lot of cassava.
I didn't know this. Which city are you in?
I'm in Kerala. So Kerala is
Oh my
likes
goodness.
cassava a lot. Yeah.
Wow. And it's not an issue. This is not an issue because it's not the same species.
Yeah, I was talking to my people around, and they were saying that we don't consume when the cassava gets bitter, we don't consume it. Like we try to harvest it like once in a year. We try to harvest it.
Yeah.
But beyond that, if it grows beyond a year, like we'll cut it off. We won't eat it.
Oh,
Mostly
that's
rats will eat it.
fantastic. That is fantastic.
I think
but-
there is this difference of the species is also an important thing. Yeah, it's a different thing that we
Sweet
use
cassava.
here.
I see.
And we boil it. Yeah, that's a very important thing.
Oh, this is
Get a
really
lot of
funny.
water.
It didn't click. Okay, to be continued. I'll see you
Yeah,
guys.
Manu, do we have a minute?
Yeah, I have a minute.
Okay, thanks,
Just give me
everyone.
1 second.
Bye.
Bye.
Let me catch you. What was your name again?
Yeah, I'm Marisol.
Marisol.
Yeah.
Yes. Do you come for lectures or?
No, I usually am not able to.
I see. You
usually
can't
not
make
able to
that
it.
time.
No, I am now. It was just like the beginning of the
I
quarter.
see.
I had like a lot of.
But you
like,
can,
Yes. I
right?
can
And you
now.
are registered in the class, right?
Yes,
Because
yes,
I haven't
yeah.
seen you.
Yeah.
Like it's a, you know, it's a very